Hey, welcome back. Here we are talking about biotech, looking at another form of biotechnology, PCR or polymerase chain reaction. Now, it used to be that if you wanted to make a copy of a gene, you had to have a cell, like a bacterial cell, copy its DNA and then divide, and then you've got two cells with the DNA. And then maybe you could have that cell divide again, and then you'll have four cells and eight cells. And it just took time to grow uh, cultures of these organisms to copy the DNA. So, finding a way to make lots of DNA much more rapidly was a huge breakthrough in sort of being able to study DNA more closely. So, with PCR, with a polymerase chain reaction, what you're doing is making lots of copies of DNA, like more than a billion copies of DNA, in less than 24 hours without using cells. Just by having a few parts present and using correct temperatures, you can actually copy DNA really rapidly. And this is probably something you'll do in undergrad. All right, so to make this work, you're going to need a fancy oven. That fancy oven is just called a thermal cycler. And here you can see the little wells where you can insert your samples and it'll make copies of the DNA for you. But in essence, what it does is it, again, takes certain like cellular machinery. Think of like DNA polymerase and primers and stuff like that. And when you heat up the DNA in certain ways, it allows you to copy the DNA without having to have all the cell. You just have certain machinery and raw materials there. And then you can make huge quantities of DNA. And that's really helpful because if you think about, like, forensic cases, you may only have like one hair or just a small smattering of blood or some tissue. And what you need to be able to do is extract enough DNA from that to make a bunch of copies. So with PCR, you only need a little bit of DNA to begin with, and then you just make a bunch of copies of whatever you need. So looking at how this stuff works, what you need to do is have a target sequence of DNA. So you're going to know there's a certain gene or segment of a chromosome that you want to copy. And then what you do is on either side of that copy, you're going to bracket it with primers. So if you recall, DNA polymerase is going to copy DNA, but it's an enzyme, so it's sort of finicky. DNA polymerase can only add new nucleotides to a previously existing strand. So what has to happen is you add these primers. So primers will be single stranded. So what they'll do is hydrogen bond with your strand of interest. So here's what happens. You have your primer and then you have your nucleotides, your A's, T's, C's, and G's. So what you do is you separate the two strands of DNA so the strands are exposed. And then the primers will attach since they're single stranded as well. And what that does is it creates a strand that has a free 3' end. So then you can use DNA polymerase to add new nucleotides. That's what's called extension here. And then you basically copy your DNA. Okay. So you need the target DNA. You need some nucleotides to make the new strands. And then you need enzymes that can operate under these special conditions. And again, if you have that and if you have correct temperatures, you can copy a lot of DNA really quickly. So the trick is look at the temperatures involved in this. 94 to 98 degrees, that's near boiling, right? This is Celsius. So uh, most enzymes, think about human enzymes, they work best at like 37 degrees Celsius body temperature. So if you take a human DNA polymerase and heat it up to almost boiling, it's going to denature, right? So how in the world do you find a DNA polymerase that can handle that sort of temperature? Well, you just turn to nature. If you look in the upper right-hand corner here, this is called the Grand Prismatic. This is a hot spring in Yellowstone National Park, and it really looks like this. It's absolutely spectacular. Now, of course, when you're there, you're down at like ground level, so you don't get that sort of view. That's sort of up on a hill looking down at it, but it's absolutely spectacular in its coloration, and it's because of bacteria that can survive in these hot springs. So Yellowstone has all these really fascinating hot springs that are really colorful because of the bacteria that survive in them. So these bacteria can handle high temps. The particular species that we use in PCR is called Thermus aquaticus. So again, these are bacteria that can survive at high temps. So it makes sense that, hey, they have DNA polymerase enzymes that can work at high temps. So Taq polymerase has a version of this DNA polymerase that can handle the heat, if you want to think of it that way, and it can extend or copy this DNA. All right, so this is a nice illustration from the book that sort of shows what goes on. There are basically 3 steps to this that just get repeated over and over again over the course of 24 hours, and you can make lots of copies of DNA. So step 1 is denaturation of the strands. Basically, what you do is you heat up the DNA. Now, remember, what's holding DNA together? Hydrogen bonds. So the first thing to break, if you heat up the DNA, you're going to break the hydrogen bonds between the strands, exposing those bases. After you do that, you cool it down a bit. I mean, this is still pretty warm, 55 to 60 or 50 to 65 Celsius. So it's still pretty warm. It's more than 100 degrees Fahrenheit, way over. But what it does is it allows these primers to attach. So again, the primers will be added in with your materials, and the primers attach to the exposed DNA strands. And then you heat it back up again a bit, and then Taq polymerase will go and copy the new strands. Once you do that, then you'll have new strands of DNA. You just cycle through again. You heat it up, you separate the new strands, you cool it down so the primer attaches, you warm it up a bit, and then Taq polymerase goes to work. It copies the strands, and then you heat it up again. So you go through this process of sort of heating and cooling, and that allows you to copy the DNA repeatedly. So here we have another animation of this to sort of help you visualize what's going on. Thank you for your patience.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling.
I think my kids must be doing homework downstairs and using the Wi-Fi. Sorry, let me try to repeat
chain
this.
Reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA.
Note the temperature.
First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
All right, that's really cool. So again, in undergrad, if you've got something that you're wanting to be studying, a particular gene, and you want to make copies of that gene, you'll put your primers and your nucleotides and your sample in the thermocycler. You let it run overnight. You come back the next day, and then you've got lots of copies of that DNA that you can run tests on. Okay. All right. So let me jump back into the presentation here. Again, I appreciate your patience with everything. Let's see. So that walks us through. So now here's a fair question. If you make all these copies of DNA, what the heck do you do with it, right? So now we're going to look at another form of technology called gel electrophoresis. So this is something you do with all the DNA that you've just copied. So what gel electrophoresis does is allow you to separate DNA or proteins based on the size of the molecules and their charge. Okay. So again, what you do is you see these banding patterns that are created. That allows you to again separate your segments of DNA based on how big the DNA is and the relative charge of the DNA. Same with proteins. So how does this work? Well, what you do is you have a gel, and then on one end of the gel you have these little wells that are created, and then you put your samples in those wells. You place your gel in a little tray or a chamber, and then you cover it with a buffer solution, and then you run electric current through this. Okay. Now, when you run current through the buffer and through the gel, DNA will start to actually move through the gel. So DNA has a negative charge with all the sugar phosphate backbone going on there with your phosphate groups. And so what will happen is the DNA will be drawn toward the positive end of the gel. So the DNA will migrate across the gel. Same with proteins. If you put proteins in these wells, they'll also migrate across the gel. And sort of how they migrate will depend on the chemical properties of the individual polypeptide. All right. So how do you separate out the strands? Well, they'll separate out based on size. So the gel that you use, this agarose gel, it has sugar fibers and then little air bubbles in there as well. So what will happen is basically the fibers and the air bubbles will prevent or slow down the movement or migration of the DNA across the gel. So basically, what happens is the bigger pieces of DNA get hung up, and so they don't move as quickly. And then the smaller pieces of DNA can squeeze their way through here, and then they move more rapidly. So the net effect of this is that you have different bands of DNA in the gel, because again, the small fragments migrate more quickly, and the larger fragments get sort of hung up. So here you see the DNA that's been cut up into these pieces. The bigger pieces get hung up and they don't move from the well as much, so you get one band. And then the smaller pieces, they migrate farther along the gel during your runtime and they create another band. So that's a really cool way to sort of separate out DNA so you can begin to analyze it. Okay. So what this does is allow us to create things like DNA fingerprinting. I don't know if you've ever heard of DNA fingerprinting before, but the thing to remember is we've all got DNA in our cells, and all of us have DNA that's unique. So if you use the same restriction enzymes to cut the DNA from different individuals, because all of us have different DNA, if you look at this, it's going to cut the DNA at different locations, right? So if we all have DNA samples and they get cut with the same restriction enzymes, all of us have samples or fragments of different sizes. So what that's going to do is create banding patterns that are unique for all of us. So that's the idea of a DNA fingerprint, is that when you digest or cut up our DNA, everyone's going to have fragments of different sizes. So everyone's going to have a unique banding pattern. So that's a really awesome way of sort of identifying individuals. And this is part of what's been able to get people exonerated from crimes that they've been convicted for, is if you have DNA samples, well, then you have a really good idea if, you know, the DNA from some crime scene matches the DNA from another individual, because you can do something like this. So there is, you know, real value in being able to do something like PCR and DNA fingerprinting through gel electrophoresis. Because if you have some small sample from some sort of like crime scene or some sort of, you know, event that needs identification, you can make copies of that and then test it and get pretty conclusive information. So here is just a little bit of practice on how to interpret or think about how to read electrophoresis. So again, the job of PCR is to make copies of DNA. So you amplify your sample. Then you cut with restriction enzymes. And after that, you can do this electrophoretic separation. You can separate the DNA out by size. So if we look here, there are the possible genotypes. If you're homozygous dominant, it'll cut in two places, so you get two bands. If you're homozygous recessive, it looks like it doesn't cut, so you get one band. But if you're heterozygous, you've got copies of both, right? So you have the two bands created by one copy of the gene, and then the single band created by another copy. So again, what that does is allow you to sort of identify DNA between individuals. Now, this is looking at just one particular gene, but again, you can look at different DNA samples from individuals. And what you're looking for is something that's called a RIFLIP. RIFLIP stands for Restriction Fragment Length Polymorphism. Again, that's a big term, but the idea is pretty straightforward. It's just that everybody's DNA is going to cut in slightly different spots, so you're going to create different sized RIFLIPs. So we all have sort of different sized fragments in our DNA due to these slight differences in the DNA. Here's another interesting example. Again, in this case, the RIFLIP, the difference is due to what's called a SNP. Okay, lots of terms here, but it's a single nucleotide polymorphism. So that means one change or one difference in the bases. So if you have a single difference in a base, that causes a different DNA sequence, and as a result of that, the restriction enzyme may not cut in that location. So here they're using the restriction enzyme DD1. So if we look, DD1 in a healthy hemoglobin gene, it cuts in 1, 2, 3, 4 locations. One big fragment and two smaller fragments. So if we look here with the healthy allele, there's one big fragment, two smaller fragments. But if we look at the second example, a person with sickle cell disease, because of this difference in the DNA, you've changed the order of the bases. Well, here the restriction enzyme doesn't cut. So because the restriction enzyme doesn't cut, rather than 3 pieces, you only get 2 larger pieces. So if we look, here's your 1, 2 larger pieces. So again, it doesn't migrate as far on the gel. So you can look at these gels and get a sense of, oh, okay, has there been a mutation in the DNA? So really powerful capacities with gel electrophoresis. All right, next thing we'll look at is gene therapy. The idea here is that you use a virus as a vector to deliver a therapeutic gene to the patient or the host organism, however you want to think of it. So again, viruses for billions of years have been sneaking their genetic information inside a host cell. And then typically what a host cell does is it copies the viral genetic information, copies the viruses explode and go and infect other cells. Well, what they'll do with gene therapy is they'll take the genes for virulence, they'll knock out the genes for virulence that would like make a person sick, and then they'll load the therapeutic gene into the viral information. And again, this is what they're doing with certain versions of the COVID vaccine. They're taking the gene for that spike protein and inserting it into the adenovirus. And then what they'll do is inject the virus into a person. And rather than it being a cold virus, I mean, the adenovirus is just a cold virus. But rather than infecting a person, what it will do is sort of sneak in the instructions for the spike protein, and then the person will, transcribe that and then produce little spike proteins, and then some of their cells will have this information, and then it will trigger an immune response. So the immune system can develop the capacity to recognize the virus. So again, it's just sort of an interesting way to get cells to produce a protein. All right. Last thing we'll look at in this segment is immunotherapy. So with immunotherapy, what you're actually doing is altering the actions of your B cells and T cells, your immune system or your specific immunity. So one thing that can happen is your immune system, we know, is on the lookout for sick cells that aren't doing well and that need to be destroyed. Well, one capacity that certain types of cancer cells have evolved to have is this ability to basically hide or switch off the activity of T cells. Okay. So if we look here, this is it's called the PDL1 pathway. So when when cancer cells are trying to evade your immune system, they'll produce this PDL1 protein on their surface. And then that basically sends a signal to our T cells to not attack, to not perform the actions of cytotoxic T's and like blow up cells. Okay. Again, here in sort of green, that's your T cell, and then gray, here's the cancerous cell. This PDL1 protein on the surface of the cancer cell inhibits the activity of the T cell. But what researchers have figured out is if you use this specialized antibody, you can sort of block the PD1 protein on the surface of T cells so that this PDL1 pathway gets shut down. And so that allows T cells to become triggered, to become active, and go and attack the cancer. So immunotherapy is promising, very promising for certain types of cancers because if certain types of cancerous tumors are using this PDL1 signaling pathway, you can actually train the immune system to sort of get past that and go and attack and destroy the cancer. All right, so we'll stop here and finish with one more segment.